So I've been obsessively going through the ivermectin toxicity studies, and what I found absolutely blew my mind. Why did so many people on social media say that this stuff was safe? It appears to be extremely cytotoxic and genotoxic, as in it's damaging to cells and DNA, and it's very likely to be carcinogenic, based on numerous different studies looking into this. Here's what I found. You can find my whole write-up on timtruth.substack.com, link in the description. Wow, so I was amazed how many studies, and I've got a number of them here, show that ivermectin is genotoxic, cytotoxic, and very possibly carcinogenic. This is horrifying. First, we're going to look at a human cell line and see the DNA toxicity and the cell toxicity. Then we're going to look at the increased mutations in fruit flies exposed to ivermectin. Then we'll look at a study of Swiss albino mice and the shown mutagenic effects of ivermectin, and the massive increase in chromosomal aberrations post-ivermectin treatment. Then we'll look at another human cell line versus ivermectin, where they find dose-dependent cell death, autophagy, and apoptotic cell death, quote, ivermectin-induced cytotoxicity. Then we'll look at a buffalo lymphocyte cell line and the increased numbers of chromosomal aberrations as the dosage increases. Next, we'll look at a mice bone marrow study and high levels of chromosomal aberrations post-ivermectin. The study, quote, reveals high clastogenic and genotoxic potential of ivermectin on mice. And this study is interesting because they looked after one dose and after a repeated dose, and they find high levels of damaged cells. Then we'll look at the Chinese hamster ovary cell line and the genotoxic and cytotoxic effects of ivermectin. And again, we see many more damaged cells post-ivermectin. Then we'll look at an ivermectin rat carcinogenicity study where they mixed in some ivermectin into the rat's diet. Very small amount, 2 parts per million. And they found an increased number of neoplasms and a statistically significant 4-fold increase in the number of pathological lesions. So what they did in this study was expose the rats who were eating ivermectin in very small amounts, and then they exposed it to a carcinogen. And they looked at the carcinogen by itself, and then they exposed it to this other group of mice who were feeding a little bit of ivermectin. And they found the rats that were eating ivermectin were hit much harder by the carcinogen. So they found an increased number of neoplasms for the group that was eating ivermectin, and a statistically significant 4-fold increase in the number of pathological lesions. Quote, in all cases, the pathological lesions were more severe in the animals receiving ivermectin and MNNG, the carcinogenic substance, compared to those receiving MNNG alone. Next, we'll look at a mosquito cell line and the genotoxic and cytotoxic effect of ivermectin, with a massive change in the damaged versus undamaged cells, whereby many more cells were damaged post-ivermectin. Next, we'll look at a study with Kebu Brahman cows, where they found cells were highly damaged and the DNA damaged as well. Significant DNA damage. Quote, the results obtained in this study demonstrate the likely genotoxic potential of the use of ivermectin in cattle. Next, we'll turn to a very interesting study about tadpole DNA and the negative effects of ivermectin, where they found genotoxic effects at relatively low concentrations of ivermectin. And as the ivermectin dosing increased, the cell viability decreased, and the DNA damage increased, and the damaged cells were more and more damaged. And finally, we'll look at a pig cell study where they found ivermectin inhibited cell growth, and they found less protein in the cells treated with ivermectin. So let's dive in. Let's start with this study by Zhang et al., looking at HeLa human cells, the Henrietta Lacks human cell line. The study is called Ivermectin Confers Its Cytotoxic Effects by Inducing AMPK, mTOR-Mediated Autophagy and DNA2 Damage. The researchers exposed human cells to ivermectin and measured and photographed the microscopy of the results. Here are a few choice excerpts from the paper. Ivermectin has significant ability to induce DNA oxidative damage and enhance autophagy in human cells, HeLa cells. Quote, the accumulation of ivermectin in animal tissues and the excretion of urine and feces into the environment is the major source of potential toxicity. Human consumption of meat or milk contaminated with livestock can result in exposure to high levels of ivermectin exposure. As expected, we found that ivermectin can induce oxidative double-stranded damage in HeLa cells, indicating that ivermectin has potential genotoxicity to human health. In addition, we observed the formation of LC3B in HeLa cells, the accumulation of Beclin-1, the degradation of P62, and the activation of AMPK-mTOR signal transduction pathway. We conclude that ivermectin produces genotoxicity and cytotoxicity by inducing DNA damage and AMPK/mTOR mediated autophagy, thereby posing a potential risk to human health. So genotoxicity is damaging of the genetic material, and cytotoxicity is the damage of cells. As shown in Figure 1a, the cell clone formation rate decreased gradually with the increase of ivermectin concentration after 6 hours of drug administration. So look at this series of photos. All right, so we have 5 different treatment groups pictured here. The first one, 0 micromoles. And they're all 6 hours in. This is a snapshot of the cell colonies, human cell line, 6 hours after the treatment of different amounts of ivermectin. So the top left gets 0 micromoles. The top right gets 2.5 micromoles. And then the 3 on the bottom get 5, 10, and 20 micromoles, respectively. And look at the difference here. There are noticeably fewer colonies for the 5 micromoles, fewer still for the 10 micromoles, and it's hard to see any because of how many fewer in the 20 micromole photo. That is stunning. So here we have it in chart format. So our control is on the left, and everything else is based off of that. So 100% is how many we saw in the control group. When they administered 2.5 micromoles of ivermectin, we see it goes down 20%. When they double that and give it 5 micromoles, that goes down to 60% of the control level. And when they double that again to 10 micromoles, it goes down even further, below 40%. And then finally, 20 micromoles, it's down below 10%. So for part A of this figure, it says, HeLa cells were incubated with ADGFP-LC3B, and then treated with ivermectin for 6 hours. The autophagosome were analyzed by fluorescence microscopy, and we see, going from left to right, 0 micromoles of ivermectin, 2.5 micromoles, 5, 10, and 20 micromoles. All the photos were taken 6 hours in, and we see on the right it's glowing much brighter, which my interpretation is that corresponds with autophagy. And here's a quick definition. This is NIH website. Autophagy, a process by which a cell breaks down and destroys old, damaged, or abnormal proteins and other substances in its cytoplasm. Then it recycles the breakdown products, and this is especially during periods of stress or starvation. So we seem to be seeing more and more autophagy as the ivermectin is ramped up. So in part B, we have a Western blot. The caption reads, after treatment with ivermectin for 6 hours, the expression of autophagy-associated proteins, LC3, Beclin-1, P62, and HeLa cells were detected by Western blot. So we have the control over on the left, 0 micromoles of ivermectin, and then we see it get darker and darker as we ramp up the ivermectin, seemingly demonstrating more and more autophagy. And we can see the same information over here for these different proteins in chart format for the different ivermectin treatment amounts. And this is relative density on the y-axis. So it's interesting that P62, the dark column on the right, is actually decreasing. And I looked that up. Apparently P62 accumulates when autophagy is inhibited. So a decreasing P62 seems to indicate autophagy. And we can see LC3 is just ramping up and up and up, dose dependent. So the paper also found that ivermectin induced mitophagy in HeLa cells. Mitophagy is the removal of dysfunctional mitochondria, and it happens through the autophagy process. So dysfunctional mitochondria, not a good sign. So this mosaic of different photos, labeled A at the top here, the caption reads, after treated with ivermectin for 6 hours, mitochondria and lysosomes were stained with mitotracker and lysotracker red, then detected by fluorescence microscopy. So here we have those same doses again, 0, 2.5, 5, 10, and 20 micromoles from left to right. The middle row is lysotracker green, the bottom row is mitotracker green, and then those are merged for the top row. And what we see is this increase in lysotracker green, and we see a decrease in mitotracker green. So it starts out mostly mitotracker, and this yields to the lysotracker for higher doses of ivermectin. Part B says after treated with ivermectin for 6 hours, the MPTP opening was detected by co-loading with calcium AM and COCl2. And then the graph, part C, the caption reads, after treated with ivermectin for 6 hours, ATP content was measured. So it seems this is their evidence to support their claim ivermectin-induced mitophagy in HeLa cells. Here's the next figure, figure 7. It says ivermectin-induced DNA damage in human HeLa cells. So for A at the top left, these 15 photos, it says anti-gamma H2AX monoclonal antibody was used to detect DNA damage, foci immunofluorescence, and DAPI was for nuclei staining. For B, the chart at the top right, relative density of gamma H2AX in the treatment of AVM for 6 hours analysis results. So on the y-axis, the percentage of colonies, and we see this ramping up and up and up. For C, it says anti-8 oxide G monoclonal antibody was used to detect DNA damage, foci immunofluorescence, and DAPI was for nuclei staining. And for this chart over here for D, where we also see it ramp up and up and up as a percentage of colonies, as the ivermectin is increased, the caption reads, relative density for 8-oxide G in the treatment of AVM for 6-hour analysis results. So let's turn to the next study, ivermectin genotoxicity and carcinogenicity, observed with fruit flies. So this study was called Genotoxicity and Carcinogenicity of Ivermectin and Amoxicillin, but we're going to obviously focus on ivermectin, in vivo systems. This was by Aparecida de Souza et al. Quote, the results revealed that ivermectin increased the frequency of epithelial tumor in D. melanogaster, considering all evaluated concentrations. Quote, findings showed an increase in the frequency of micronuclei in T. pallida, treated with 11, 23, and 46 times 10 to the negative 5 micromoles of ivermectin. We conclude that chronic exposure to ivermectin is directly associated with events resulting from genetic instability, genotoxicity, and carcinogenicity. Ivermectin also appears to drastically reduce survival rate of fly progenies as the dose is increased. So you can see the fly population goes way down as the ivermectin goes up. This is measured by the survival percentage in this chart. And over here on the right, after we get by the two controls, this is ivermectin as the dose is increased, and we see the drop-off in the survival percentage. So let's look at the tumors that they noted in these flies. So for each of these treatment groups and for the control, they analyzed the number of tumors, and I found the results extremely startling. So the first graph is an aggregate, and then we'll break it down by category. So this chart shows us total tumors based on the ivermectin dosage. And we see this really frightening line by which as they increase the ivermectin dosage, this is measured in micromoles, the number of tumors increases. So this is a really concerning chart. This shows us the total tumors in these fruit flies as the ivermectin dosage increases. So it's causing physical abnormalities, it would seem. So for all these charts, we put it on a log scale, and these data points all line up. This is a logarithmic regression line, and we see a very concerning pattern that as the ivermectin is increased, we see an increase in the total number of tumors. Extremely concerning. So here are the 6 categories of tumors that they tracked. Eye tumors, head tumors, wing tumors, body tumors, leg tumors, and halter tumors. And again, each of these charts are logarithmic scale. The regression lines are logarithmic. And we see this extremely concerning pattern where more and more tumors are observed as the ivermectin goes up. So really concerning, we have the tumors of the eye increasing with ivermectin. We have the head tumors increasing and increasing as ivermectin is increased. We have the wing tumors really close to the regression line, all the data points. Then we have the body tumors showing some really concerning chart action. Here are the leg tumors and then the Halter tumors. And a quote from the paper, "It was observed a dose dependence in the frequency of micronuclei, which is a structural abnormality of a cell. In T. pallida, considering the highest concentrations, 11.42, 22.84, and it mentions the ivermectin dosage differing statistically from the negative control, evidencing a genotoxic effect of ivermectin. So the results observed showed that ivermectin can increase the damage in the genetic material, leading to genetic instability." And this is an interesting concerning chart, micronuclei per 100 as ivermectin increases. So ivermectin on the x-axis, percentage of cells with micronuclei on the y-axis. And as the ivermectin increases, so too does the rate of micronuclei. Very troubling. So why fruit flies, we should wonder? Well, the authors explain as such. In D. melanogaster, which is the common fruit fly, the test for detection of epithelial tumor represents one of the most promising alternatives to evaluate and identify possible carcinogens. This test has also been used to verify carcinogenic events of different substances. The next study looks at ivermectin mutagenicity in Swiss albino mice. The study is titled "The Mutagenic Effects of Ivermectin in Germinal Cells and Serum Protein of the Mouse." A quote from the paper, "Ivermectin is extremely toxic to fish and aquatic life. Some animals showed reduction in the fertility, the number of variable fetuses, and sperm count following treatment with ivermectin." Quote, "Analysis of the treated samples revealed significant increase in meiotic aberrations." So it goes up to 33.84% for meiotic aberrations versus 5.8% for the control. These findings support the mutagenicity of ivermectin. And here's a chart showing how substantial that jump is. Abnormal metaphases, 5.8% for the control. And with the ivermectin that they gave, it goes up to 33.83% abnormal metaphases as a percentage. That is a substantial leap. That is a massive jump. So the paper breaks it down by different category of chromosomal aberration. And in all the categories, we see a massive increase with the ivermectin versus the control. You can see that in these 8 charts on the screen. So we have AU chromosomal aberrations, XYU chromosomal aberrations, AU plus XY chromosomal aberrations, RIV chromosomal aberrations, CIV chromosomal aberrations, CVI chromosomal aberrations, F chromosomal aberrations, and polyploidy chromosomal aberrations. Just take a second and look at these huge differences. So for AU chromosomal aberrations, there were 0.5 in the control group, and there were 6 in the ivermectin treatment arm. That's an increase of 1100%. And then for XYU chromosomal aberrations, we had 1.6 in the control arm and 11 in the ivermectin treatment arm. That's a 587.5% increase. In the AU plus XY chromosomal aberration, there were 0.3 in the control arm, and there were 1 in the ivermectin arm. That's a 233% increase. In the RIV chromosomal aberrations, we had 0.16 in the control, 0.83 for ivermectin. That's a 419% increase. For CIV chromosomal aberrations, 2.16 in the control, 9.3 in the active arm. That's a 330.6% increase. For the CVI chromosomal aberrations, there were 0.16 in the control, 1.83 in the ivermectin arm. That's an increase of 1,044%. For F chromosomal aberrations, there were 0 in the control arm, 0.16 in the ivermectin arm. And for polyploidy chromosomal aberrations, there were 0.83 in the control and 3 in the ivermectin arm, meaning a 261.4% increase. Wow. Here's another quote from the paper. Effects of ivermectin on spermatocyte chromosomes. Spermatocytes of the treated samples revealed significant increase in chromosome aberrations over the control values. Quote, the present observations pointed to the mutagenic effects of ivermectin. The frequency of translocation is significantly higher than that found in the control samples. So let's turn to the next study. Ivermectin versus human neuroblastoma, SHSY5Y study. The study is called Ivermectin-Induced Apoptotic Cell Death in Human SHSY5Y Cells. Involves the activation of oxidative stress and mitochondrial pathway and AKT-mTOR pathway mediated autophagy by Zhang et al. Quote, the results show that ivermectin treatment for 24 hours could induce dose-dependent cell death in SHSY5Y cells. Compared to the control, ivermectin treatment significantly promoted the production of ROS, mitochondrial dysfunction, and cell apoptosis. Ivermectin treatment also promoted mitophagy and autophagy, which were characterized by the decreased expression of phosphorylation, pAKT, and pMTOR proteins, increased expression of LC32, BICLIN-1, ATG5, PINK, and PACIN-1 proteins, and autophagosome formation. Our results reveal that ivermectin could induce autophagy and apoptotic cell death in SHSY5Y cells. Quote, ivermectin-induced cytotoxicity is dose and time dependent. At 6 hours and 12 hours, ivermectin treatment at 15 micromoles significantly decreased the cell viabilities to 44.3% and 35.6%, respectively. At 24 hours, ivermectin treatment at 0.625, 1.25, 2.5, 5, 7.5, 10, and 15 micromoles decreased the cell activities to 98.5%, 92.4%, 81.9%, 70.2%, 51.3%, 37.6%, and 23.8%, respectively. Correspondingly, marked cell morphology changes, including a spindle-like cell body, shrinkage, and dendrite fragmentation in high concentrations of ivermectin. At 7.5, 10, and 15 micromoles for 24 hours respectively, were also detected. So here's a concerning chart showing us cell viability normalized to the control levels of these different amounts of ivermectin. 6 hours in, 12 hours in, and 24 hours in. So we get 3 snapshots. And it's organized so that the smallest dose, 0.625, is on the left, moving to the largest dose, the blue, 15 micromoles of ivermectin on the right, of each of these 3 time points. And we see a huge drop-off, and it's dose and time dependent. So for example, for the 15 micromoles, just look at the 3 blue lines. You can see it's still decreasing over time. So a big hit already 6 hours in, but it's not done. Same for many of the other doses. And here's another chart from the paper. The red is the viable cells. The yellowish green is the early apoptotic cells. And the green is the late apoptotic/necrotic cells. So you can see the good cells, the viable cells, decreasing as a percentage. Early apoptotic cells rapidly increasing as the ivermectin is increased. And we also see an increase in the late apoptotic/necrotic cells as the ivermectin is ramped up. The next study is going to look at ivermectin in buffalo peripheral lymphocyte cells. The paper is called Anti-Mutagenic Activity of Some Natural Substances on Ivermectin Genotoxicity in Lymphocytes of Buffalo by L. McAway et al. So the first chart looks at cell abnormalities. We have 3 categories here. Binucleated lymphocytes shows a decrease over time. So I'm not sure exactly how to interpret that. These other charts look much more concerning. The second chart, binucleated lymphocytes with micronuclei versus ivermectin. So we have 3 data points with the line. The x-axis is how much ivermectin, and the y-axis is how many binucleated lymphocytes with micronuclei. So that's a type of cell abnormality. As is this over on the right, micronuclei versus ivermectin. So on the x-axis, ivermectin, on the y-axis, number of micronuclei. And we see this stark increase with the ivermectin amount of the micronuclei observed. This is a very concerning chart. This shows us various categories of structural chromosomal aberrations. On the x-axis, how much ivermectin was given. So we see for all of these categories, The blue is gaps. The red is breaks. The yellow is deletions. The green is fragments. And the orange is centromeric attenuations. All increasing as the dose increases. That's frightening. It says, quote, ivermectin-induced dose-dependent significant increase in the number of binucleated lymphocytes with micronuclei, and also the frequency of total chromosomal aberrations. In addition, the numbers of binucleated lymphocytes showed dose-dependent decrease than control. These results revealed that the drug has a cytotoxic effect on the number of cell divisions, as the micronuclei are small chromatin-containing bodies, arising from chromosome fragmentation by breaks or deletion. The results of micronuclei formation confirmed our results of chromosomal aberrations, indicating clastogenic effects of ivermectin. And according to Wikipedia, clastogen is a mutagenic agent that disturbs normal DNA-related processes or directly causes DNA strand breakages, thus causing deletion, insertion, or rearrangement of entire chromosome sections. The next study is going to look at mice bone marrow in vivo with ivermectin. The study is called The Cytogenic Potential of Ivermectin on Bone Marrow Cells of Mice in Vivo by Sweefy et al. Quote, ivermectin induced a high level of chromosome aberrations in somatic cells, as it is ascertained by chromosome aberration assay and micronuclei production in bone marrow cells. This study revealed high clastogenic and genotoxic potential of ivermectin on mice. So the first experiment was a single injection, and the second experiment was a repeated injection. Quote, a single injection of ivermectin resulted in a significant increase in percentage of aberrant cells. And they tracked 6 different categories, and then there's a combo category if a cell had 2 or plus aberration types. So we have these different cell aberrations, chromosomal aberrations, plotted as a function of days on the x-axis. We have a regression line. Those are the slanted, thin red lines. And then we also have the control level. That's the transparent red horizontal lines. So here's after the first injection of ivermectin, chromatid or chromosomal gap aberrations. So some of the data points in the first 3 days are under this control line, but day 7 well surpasses it. The next category tracked Robertsonian translocation aberrations. This time, the first 3 days well in excess of the control line, but these aberrations drop off by day 7. The next category, end-to-end association and/or ring aberrations. All of these data points well in excess of the control level and trending upwards. That's a startling chart. The next category, eccentric fragment and/or break aberration. With the exception of day 2, all of the points are in excess, some drastically in excess, of the control line. On day 2, it's right up against this line. The next category, dicentric chromosomal aberrations versus days. We see a huge spike on days 2 and 3 here. Day 1 is a little bit under the control. Day 7 right above it. In the last single category, deletion chromosomal aberrations. On day 1 is right up against the control, but then for days 2, 3, and 7, well in excess. Big spike in deletion chromosomal aberrations. In the final chart tracks cases where there are 2 or more aberration types. We can see day 1 and 2 right in line with the control, day 3 in excess, day 7 is under the control line. They also looked, after the first injection of ivermectin, abnormal metaphases, with gaps and without gaps. So on the left, abnormal metaphases with gaps, on the right, without gaps. These are well in excess of the control lines, which you can see here, these horizontal lines down below, in red, that's the control level, and these red dots are the observed data points. Much, much higher than the control. But it gets worse with repeated exposure, as we see in the second experiment from this paper. 2 injections of ivermectin. So let's look at this chart. So before we go category by category, let's take a bird's eye perspective. This is looking much worse than the first chart. We see these dots well in excess of the control lines, these horizontal red lines. Most all of the dots are above these lines, with 3 exceptions. So let's go category by category. The first category, chromatid and/or chromosomal gap. Big spike on days 1 and 2 after the repeated injection of ivermectin. It decreases a bit right in line with the controls for the remaining data points. Robertsonian translocation. In the first week, it's well in excess of the control, and then it drops down right in line. with the control levels. The next category, end-to-end association and or ring aberrations. This one is frightening. It skyrockets right out of the gate on days 1, 2, and 3. The highest point is day 7, and then it falls a little bit, but still very, very elevated. The next category is acentric fragment and or break aberration. All 5 data points well in excess of the control level, as you can see. The next category, dicentric chromosomal aberrations. We see a few data points well in excess on days 1, 2, and 3, and then it drops off and actually under the control line on day 7 and day 14. In the final category, before we get to 2+, deletion chromosomal aberrations. Look at this. Much higher than the control level on days 1, 2, 3, 7, and 14. And this one's really concerning too, and might be why we saw some of the data points underneath the control line, because of how many cases now we're seeing of 2+ aberrant types, which would all end up on this chart. So we have on day 1, 2, and 3 much higher levels of cells with 2+ aberration types. On day 7, it's still well elevated, and on day 14, massively elevated. So trending upwards, very, very concerning. And that could be why we're seeing some of these data points underneath the line. And again, just like before, we also look at the abnormal metaphases, on the left with gaps, on the right without gaps. Both series of data well in excess of the normal control levels, as you can see by these red lines, and the red dots well above them. So this is a concerning aspect here. These are the damaged cells over time, post-ivermectin dosages. On the left after the first dose, the single dose. On the right after the second repeated dose. And we see these high levels of percentage cells damaged. So we're seeing these very high levels of percentage cells damaged, especially after the repeated dose, especially in the first few days after the injection. So very concerning. It hits almost 60% damaged cells on day 2 after the repeated dose. You can see it decreases a bit by 2 weeks out, but it's still over 30% after the first dose, and it's over 40% at 2 weeks in. 2 weeks after the second dose, it's at 40% damaged cells. I find this horrifying. Okay, so the next study used Chinese hamster ovary cells. The study was called In Vitro Genotoxic and Cytotoxic Effects of Ivermectin on Chinese Hamster Ovary Cells, CHOK1 is the cell line, by Molinari et al. And they found that ivermectin caused DNA strand breaks in the cell line. So we have 1, 5, 10, 25, 50, 100, and 250 micrograms per milliliter of ivermectin. So one thing that really jumps off the chart here is once it hits 50 and then 100 and 250, it says not determined since a complete cellular death was achieved in cultures. So that seems to speak towards the cytotoxic effect of ivermectin. So I charted out some of this information. A decrease in mitotic index typically indicates genotoxicity, I hear. Quote, ivermectin exerted a cytotoxic effect of CHO cells within the 25 to 250 micrograms per milliliter concentration range. Quote, the highest ivermectin concentrations, 50 to 250 micrograms per milliliter, resulted in cellular cytotoxicity, clearly revealed by delaying the cell cycle progression, decreasing the mitotic activity, and inhibiting cell growth. Both chemicals induced DNA strand breaks, revealed by the COMET assay. A brief 80-minute pulse treatment of between 5 and 50 micrograms per milliliter of ivermectin or 25 and 50 micrograms per milliliter of Ivomec resulted in a manifest level of single DNA strand break induction. And then we have this chart, which is very concerning. So the green line at the top is the undamaged control. So without ivermectin, 80% undamaged cells. The blue line shows us the ivermectin data. On the x-axis, ivermectin micrograms per milliliter. And we see the undamaged cells diverge drastically from the control group. More and more so, the more ivermectin. And we see that in terms of the elevated, slightly damaged cells. So you can see the amount that the yellow line is over the teal line are the additional slightly damaged cells seen in the ivermectin arm of the trial. And it increases as more ivermectin is given. And then it was nearly 0 damaged cells in the control. That's the purple line at the very bottom at 0%. And then you see the red line, which is the ivermectin arm of the trial. Damaged cells. Now we're starting to see those. Once the ivermectin's added, we start seeing damaged cells. Cytotoxicity. The next study by O'Connor et al. Is interesting. I was only able to find the abstract. The study is called Increased Pathology Incidence in the forestomach of rats maintained on a diet containing ivermectin and given a single dose of MNNG, which is a carcinogen. So basically they had two groups of rats. One was given the ivermectin, the other was not given ivermectin, and then both groups were exposed to a known carcinogen. And they measured things like tumors and pathological lesions between the two groups. And it turns out the group that was taking ivermectin in their diet, 2 parts per million, which seems really small, had, quote, an increased number of neoplasms. So it says 9/26 versus 3/18, which if I'm reading this right, that means on average 0.35 neoplasms per rat in the ivermectin group versus 16.7 on average neoplasms per rat in the non-ivermectin group. So that's more than double the rate, with just a little ivermectin in the rat's diet. And it goes on, quote, a statistically significant 4-fold increase in the number of pathological lesions. So there were 18 of these pathological lesions in the 26 rats in the ivermectin arm, which is on average 0.69 per rat. And then in the no ivermectin arm, 3 pathological lesions for the 18 rats, which is on average 0.167. So that's a massive increase, on average, in the ivermectin versus the control arm. The average number of pathological lesions per rat increased over 4 times when the rats were eating food laced with 2 parts per million ivermectin. So very, very small quantities seem to make a huge difference. And it says this includes pre-neoplasia in the forestomach. In all cases, the pathological lesions were more severe in the animals receiving ivermectin and MNNG compared to those just receiving MNNG alone. The next study looks at mosquito cells in vivo. So the study is called Genotoxic and Cytotoxic In Vitro Evaluation of Ivermectin on AIDS albopictus larvae CCL126 cells. So these are mosquito larvae cells. It says ivermectin induced DNA strand breaks, enhancing both slightly damaged and damaged cells at 25 to 50 micrograms per milliliter ivermectin. Quote, cytotoxicity was observed at concentrations higher than 25 micrograms per milliliter ivermectin. Ivermectin exerted a delay in CCP and a reduction in PRI when 25 grams per milliliter was employed. Quote, cytotoxicity was observed at concentrations higher than 25 micrograms per milliliter ivermectin. Ivermectin exerted a delay in CCP and a reduction in PRI when 25 micrograms per milliliter was employed, whereas cytotoxicity was observed at higher concentrations than 50 micrograms per milliliter. A marked reduction of about 98% of the mitotic index compared to controls was noted with 25 micrograms per milliliter of ivermectin. NR and MTT assays revealed that ivermectin induced a cell growth inhibition within the 1 to 250 micrograms per milliliter concentration range. Data indicated that ivermectin exerts both genotoxicity and cytotoxicity in insect cells in vitro. Okay, so here's a very interesting chart that goes over this data. You can see these horizontal lines are the controls, and then the arrow indicates the direction that we see once the ivermectin is introduced. And on the x-axis is the dose of the ivermectin. The y-axis is percentage. So we see the percentage of undamaged cells drops massively from the control group to the ivermectin, and it drops further the more ivermectin is introduced. This is a massive drop-off, a massive decline, nosedive, in the undamaged cells once ivermectin comes into play. The other two categories increased, concerningly. So we have the slightly damaged cells increasing from this baseline yellow line here to this slanted incline. Yellow line with the 3 dots showing us the data points. So the more ivermectin, the more, quote, slightly damaged cells. And then also we see the damaged cells in red. We see the baseline control group right around 0%. And then we see the ivermectin at higher levels causing damaged cells, showing seemingly a cytotoxic effect. We also see a big drop-off in the mitotic index, which typically indicates genotoxicity. So why did they use insect cells? This is a quote from the paper. It was demonstrated that among insects, mosquito cell cultures provide a useful in vitro system for studying deleterious effects induced by xenobiotics. Investigations demonstrated that DNA damage induced by different chemicals was found to be less extensive and repaired more efficiently in these mosquito larvae cells compared to Chinese hamster ovary cells. So the next study looked at ivermectin in SIBO Brahman cows in vivo. The paper is called Comet Assay to Determine Genetic Damage by the Use of Ivermectin in zebu cows. The paper says, quote, "The values of classification of comets indicate cells with high levels of damage. Grade 3 is cells with high damage. The rate of DNA damage of the treatment to 1% to 3.15% was significant. The results obtained in this study demonstrate the likely genotoxic potential of the use of ivermectin in cattle." Quote, "Regardless of the ivermectin concentration, the presence of nuclei with DNA migration was observed at a percentage greater than 75% in all cells observed per plaque, demonstrating the ability of ivermectin compound to produce simple chain breaks in the DNA molecule. The COMET classification measures how damaged the DNA is. All of the no treatment control group measured a 0 on the COMET scale. So there were 5 cows in the no treatment group, 5 cows in the 1% ivermectin group, and 5 cows in the 3.15% ivermectin group. All 5 in the control group measured 0 on the COMET scale. So the COMET scale, I believe, is 0 to 4, with grade 3 meaning cells with high damage. Grade 4 would be even higher than that. So you want to score a 0, and all the control group cows measured 0. Here are the other 10 cows. So the first 5 got the 1% ivermectin, and the second 5 get the 3.15% ivermectin. So we see in each group 4 get grade 3 classification on the COMET scale, and then 1 in each group grade 4 COMET classification. Very concerning, the degree of damage to the DNA. Quote, "The results found in the present study constitute concrete evidence for the induction of genomic damage as exerted by ivermectin using the COMET assay methodology." Quote, "Ivermectin decreased glucose utilization in IBRS2 cells in 11, 30, and 31% respectively after 24, 48, and 72 hours. The next study pertains to tadpoles and measures the change in the DNA after ivermectin. The study is called Genotoxicity of 3 Ivermectins on Polypodetes megacephalus tadpoles using the COMET assay by Gang et al., which found, quote, "genotoxic effects at relatively low concentrations." Our first chart shows a dose-dependent decline in cell viability as ivermectin dosing increases. We see it just continue to drop off. As the ivermectin goes up, cell viability goes down. From the paper, quote, "Our results showed clearly that ivermectins caused dose-dependent DNA damage on amphibian tadpoles. The 3 ivermectins increased the DNA damage observed in the tadpoles in a dose-responsive manner. There were strong linear correlations between the DNA damages and the concentrations of the 3 test substances. The cellular distributions of DNA damages in tadpoles are shown in Figure 3. Of the tadpoles treated with increasing concentrations of the 3 test substances, higher proportions of cells had greater amount of DNA damage than those of the negative control. And we also see DNA damage frequency increase as ivermectin is administered in higher doses. Very concerning. The more ivermectin, the more DNA damage. Figure 3 shows us the distribution of DNA damage based on damage class of DNA patterns. So again, we have class 0, class 1, class 2, class 3, and class 4. And what we see, class 0 is where you want to be. Little DNA damage. That's where most of the cells lie when there's 0 ivermectin. But this drops off drastically. Fewer and fewer in class 0, and more in class 1, class 2, class 3, class 4. And as we increase the ivermectin, more of these later classes, indicating high levels of DNA damage. So one more quote from this paper. "According to these results above, and our finding that ivermectins can cause DNA damage in tadpoles at the concentrations below the recommended applied levels, we consider it possible that ivermectins are carcinogenic and confirm it has the negative impact on the development of tadpoles." And one last study here, ivermectin in pig cells. The study is called Toxicity Assessment of the Antiparasitic Ivermectin. Quote, "Our data show that ivermectin inhibited cell growth. This effect was time and dose dependent and ranged from 38 to 84% after 72 hours in cells treated with 2 to 20 pg per ml. At the dosage of 40 pg per ml, cell death occurred within 24 hours. They also found that protein synthesis is inhibited in a continuous way in the cells exposed to ivermectin. When compared to the control cultures, the protein of treated cells is 13, 24, and 28% less, respectively, after 24, 48, and 72 hours. So I find this all extremely disturbing. I have to tell people, because this is being peddled as a miracle cure. I'm not saying that, but a lot of people are saying it's a miracle cure. It's safe and effective. And I've been a little bit remiss to not uncover this until now. So I'm making up for lost time. I'm trying to sound the alarm. I don't know why this is being pushed on us. Why were we told that this was safe? This is a huge scandal and a massive betrayal, if you ask me.